All right, well, hi, thanks for having me. My name is Robert Langefeld. Today I'll be presenting on pyGEMMA. pyGEMMA is an implementation of linear mixed models. It was originally designed for genome-wide association studies. It's used in Python or R and implements a number of optimizations to run faster than most competing methods for fitting linear mixed models in these kinds of analyses. My clicker is broken. I'll give a little bit of background about myself before I jump into things. Currently, I'm a PhD student in biostatistics here at the University of Michigan. Before that, I got my master's at the end of the summer. And before that, I was at Wake Forest University and I got a BS in mathematics and computer science. Research-wise, I do statistical genetics and right now more spatial transcriptomics work. I do deep learning and high performance computing focused methods in those areas. Outside of research, I do Taekwondo at University of Michigan State. I like learning languages and I play classical guitar. Figured out a little fun there.
Did you bring?
I did not bring the guitar. I also want to start by thanking my lab, specifically Dr. Shang Zhou, who is my advisor for my PhD, and Boran Gao, who just graduated and is now an assistant professor at Purdue. So I appreciate both of their help on this project. Before I dive into all the math, I want to start with an overview of what these types of studies are. So if we look around the room, you'll notice everyone has different heights, eye color, hair color, and other unique characteristics, right? And below the surface, we also have different conditions like diabetes or predispositions to certain injuries. We're all a little different. These observable characteristics are what we call traits. And traits are influenced by a mix of our environment and our genetic makeup. Talking about the genetic side of things, our genetic code is like a massive instruction manual for building and maintaining our bodies. The manual is written in a language of A, T, C, and G. And these letters stand for adenine, thymine, cytosine, and guanine. These are the building blocks of DNA. Think of DNA as a twisted ladder where these letters pair up to form the rungs. So A pairs with T and C pairs with G. The sequence of the letters determines everything about us. The specific sequences we call genes code for proteins that perform various functions in our bodies. Sometimes a single change in this sequence, known as a single nucleotide polymorphism, or SNP, can affect how proteins work and consequently influence a trait. For example, a tiny change in a gene might increase someone's ability to gain muscle mass. So how do we read this instruction manual? That's where sequencing comes in. Sequencing is the process of determining the exact order of these letters in a DNA molecule. Thanks to advances in technology, we can now sequence entire genomes quickly and affordably. This allows us to identify genetic variation that might be linked to a specific trait. This brings us to genome-wide association studies, or GWAS. These studies scan the genomes of many individuals to find genetic variation associated with a particular trait. By comparing the DNA of people in a large sample, researchers can identify SNPs that are more common in individuals with a specific trait. This helps us pinpoint regions of the genome that may influence the trait and provide insight into the underlying biological mechanisms. To conduct a GWAS, we take a sample of individuals from the population. DNA is taken from each of those individuals and sequenced. This gives us information on the configuration of each person's genetic code at a given SNP. We code these based on how many copies of the non-reference version of that SNP each individual has. We can then fit a linear model to compare the number of copies to each phenotype. So we can see positive relationships or negative relationships or even no association at all. We test the significance of the slope coefficients from these models for millions of SNPs across the genome. The negative log 10 of these p-values is often visualized in what's called a Manhattan plot, which I'm showing here. And spikes in these plots represent regions of the genome where we see an association with our trait of interest. This identifies candidate SNPs as variants warranting further examination. In basic linear regression models, we require individuals to be independent, but we recognize this is not how biological inheritance works. We get half of our DNA from our mother and half from our father, and share approximately 50% of our DNA with our siblings. As we extrapolate that further, we notice we're at least somewhat related, at least distantly, to almost everyone, leading to similar genotypes. Since genetics can strongly influence traits, we see correlations in traits between related individuals and genetically similar individuals as a result. Research has historically observed that individuals in similar geographical regions and those with shared ancestry also cluster when principal component analysis is applied to their genomes. All this evidence motivates us to develop models that can account for correlation among related individuals. I could start here trying to explain the math behind these methods, but I thought it would be better to start with a graphical example. So here we'll start with the basic components of our model. The phenotype is our trait of interest, say height. The SNP matrix contains a column for each SNP that we're interested in. The covariate matrix may contain several covariates like sex or age, but often will contain several principal components. This strategy has been shown to help account for population stratification. Lastly, the genetic relatedness matrix is a normalized measure of how genetically related our individuals are. For a given SNP, we want to formulate this into a linear model. Here, we're just going to use a basic linear regression model. We'll introduce coefficients for our SNP and for the covariates. We'll also add random effect terms or random intercept and random error term to make this a mixed model. We structured the random terms so that the random intercept accounts for phenotypic correlation among related individuals, and the error term captures environmental variation and other forms of error. Here, tau inverse is the variance of the residual errors, and lambda is the ratio of genetic variation to the environmental variance. This parameterization helps us fit the model much more efficiently from a computational standpoint. When we compute our relatedness matrix, we do it as x transpose over p, where p is the number of SNPs. X is our column-wise centered and standardized SNP codings. Remember, these SNPs are coded as 0, 1, or 2. So this means that our relatedness matrix corresponds to the average genetic covariance between any two individuals. So the diagonal of this matrix will always be 1. This is what the model looks like when laid out mathematically. But here we're specifically interested in estimating and testing the beta coefficient, that beta G, so that we can determine the genetic effect on our phenotype of interest for each SNP. There are several other methods that exist for fitting these kinds of models. Each of them has their own flavor and way of doing things. We'll focus on GEMMA, since that's what pyGEMMA is designed to replicate, but we compare against a lot of these different methods. So GEMMA is an efficient method for fitting linear mixed models for GWAS. It was written in C++, uses custom BLAS functions for fast linear algebra, and works with a number of common data formats. However, it can't handle generic data formats, so there's no custom file reading that you can introduce. There's no SNP level parallelization, and it requires a command line interface, which can be challenging for people who are comfortable in Python or R. There's no plotting functionality built in, so it lacks that, and it's also limited by the sample size it can run. It starts to struggle past 20,000.
20,000 SNPs or
20,000 individuals. pyGEMMA is a faster implementation of GEMMA. I recommend it because it scales to the hardware and data, so it maintains its performance even when we have moderate or large sample sizes. It has a much more efficient algorithm, including memory optimizations for faster computation and an underlying design that's built for high performance computing environments. It's also easy to use in Python and R. It's a single function to fit the linear mixed model and has built-in plotting functions for you. Breaking down those optimizations, first step we take is we parallelize over SNPs, and we use shared memory to make this possible. Second, we optimize the memory layout itself and use 3D tensor data structures for optimal storage. Third, we optimize the linear algebra using BLAS and LAPACK and removing redundant operations, which allows us to be faster and more efficient. Lastly, we use high performance computing ideas to scale it to near biobank data. I'll go into each of these a little further. To parallelize and over the number of SNPs, we're going to run one SNP on each core. This means our speedup is going to be approximately linear with each core that we add. So for each CPU, we'll just assign one SNP, copy it over. This gets extrapolated a little further, but this is the basic idea. When we fit this model, the original way we would have to do this is copy the relatedness matrix over to each of these processes that's been spun up on each core. But that's a rather expensive way of doing things because this thing can be large, can be up to a terabyte or two. So what's a better way to do this? Well, you can use something called shared memory. If everything's on one node, you just give each process a pointer to the original memory location, and on the operating system side, you open that up so that it can access that original point, which means you just don't make a copy. So they can all access this without incurring overhead. So overall, to summarize that section, the parallelization scales the runtime based on how many cores you use. So the more cores you have, faster you can run, more computation in less time compared to what GEMMA was doing. Then we just aggregate our results from each of the processes after we finish. We use shared memory to allow us to keep those gains without copying the matrix. And this means our memory footprint remains relatively low. Not exactly what it would be if you just did one process, but still pretty close. Moving on, we had to optimize the memory layout because otherwise pyGEMMA would still be slow. The likelihood computations were fine when you do it one time, but we have to do this in an optimization scheme and then for millions of SNPs. So that's rather expensive. Thus, we have to figure out how to optimize within a single SNP. Breaking down the method a little further, the way we estimate this mathematically, we're going to find the maximum likelihood estimate or restricted maximum likelihood estimate of the lambda parameter. And then we can use that to compute our tau, beta, and the variance of beta estimates. And we use that to conduct a wall test of that beta coefficient. And we do that once for every SNP. I'll give a little background about the parameterization. Don't get scared away by the math. But we'll call G the covariance between the random effects. So this has to do with that relatedness times our Z, which is our random design matrix. H is going to be our overall covariance with the tau inverse divided out. So you can see the lambda parameter still in there. Within our likelihood evaluations, we'll need to note these P matrices. They will just become important, but you can notice they're rather difficult to compute. And we can also go through and define a recursive relationship for these P matrices based on the covariance. So we need from 1 to C plus 1, where C is the number of covariates that we have. And then we define Px to be P C plus 1. This gives us a really nice structure to work with. In terms of how you compute these, it'll become clear how these are relevant in a second.
Those are projection matrices, is that right? Mm-hmm. And what are the dimensions of them? Are they large or are they just covariate size?
C plus 1 by C plus 1. Okay. No, I take that back. And by n. Yeah.
Where n is the number of individuals?
Yes. Sorry. So you'll notice that when we look at both restricted log likelihood and a modified version that allows us to do better optimization, these P matrices and the H matrix all show up everywhere. We need efficient ways to compute them because of that. Additionally, they show up in the score function and in the derivative of the score function. Because we need to do these operations millions of times, we're going to use eigendecomposition to get around some of this. So eigendecomposition is we're decomposing it into this U lambda U transpose. And we're doing so in such a way that U transpose is U inverse. There's a lot of math here, but these are just some of the operations that show up in likelihood, the score computations that we require, that we can optimize once we have our eigendecomposition. So if we want to invert that H matrix, we're able to do so just using those operations and vectorizing it, because we can just flip the eigendecomposition on its head. If that makes sense. For example, trace becomes pretty easy, determinant, all these operations become very nice once we work with that eigendecomposition. We also pre-multiply y, x, and w by u transpose so that we're removing redundant operations, and we just pre-multiply at the beginning, and then everything works so we can pull out the u transpose from a lot of these. Yeah.
Does that third bullet say that? Oh, okay, I see a comma now. Okay, never mind, thank you. Okay.
I'm so sorry. Yeah, I should put it on a different line.
I didn't see the comma.
So moving a little further, the way we actually find these optimizations is we break the lambda parameter into intervals on the log 10 scale from 10 to the negative 5 to 10 to the 5. And then we apply Brent's algorithm within each of those intervals and optimize it for at most 100 iterations, guaranteeing a relative error of 10 to the negative 1. Then we apply Newton-Raphson starting from Brent's result for up to 100 iterations with a relative error of 10 to the negative 5. Reason we do this is Brent's method only requires a score function. Newton-Raphson requires a score function and the derivative. The derivative of the score function is not nice to compute for this, so we don't want to do that. Returning to our optimized layout, we have to use these P matrices for a lot of calculations. It's required also, in addition to just the likelihood and partials, for estimating our beta coefficient, alpha, tau, lambda, all these things would be expensive if we didn't have a better way to compute these subresults. When we look at memory in a computer, memory is stored linearly. So in this diagram, the number here represents the actual order of memory. So when we think about how memory stores it, it stores it as, or how GEMMA stores it, it stores memory as a matrix for storing all these subunits. So it actually looks like this. CPUs need to access elements when they perform what's called a fetch call. So they go pull a chunk of elements out of memory. So this means for this data structure, in order to pull P1 out, we're going to need to perform 3 operations. 1 to grab the first row, 1 to grab the second row, and 1 to grab the third row. That's 3 operations, and that gets really expensive really fast. There are a couple ways that we can minimize doing these memory accesses. One, we could use the PIs only when storing them. This causes issues because dependencies between some of these PIs don't play nicely. So that's not an option. Option 2, we could store the matrices separately in memory, but that's not optimal since sometimes the fetch calls overlap and we could reuse them, but we're not. Better way to do it would be tensor-style memory structures that are optimized when we need ordered submatrices. That's what we go with here. So tensors are just multi-dimensional matrices. The 0-dimensional tensor could just be a scalar. 1-dimensional tensor is vector. 2-dimensional tensor is a matrix. 3-dimensional tensor is a cube. I don't go higher than that here because that's all we're using, but it can generalize further. We can use that to minimize the number of memory accesses by structuring it this way as one of the cubes. When we do that, you see we can pull P1 out with just a single memory access. And that's really nice because it means we've just done, instead of 3 operations, 1 operation. But as you scale covariates or other things like that, suddenly that gets really expensive because you can only pull a certain amount out of memory at a time. Additionally, these are aligned very nicely in memory, which means BLAS and LAPACK operations can be applied directly to them. And it's much faster than for loops to do that. We're not often interested in just these PI matrices. We would rather jump and calculate something like these matrices of the form A transpose PIB, where A and B are either one of the covariates or Y or our genotype. I'll also note that just due to how symmetry works out, A transpose PIB is also equal to B transpose PIA. So that's a nice property that we get to work with. We want to vectorize these operations. So we define T as a three-dimensional tensor that is C by 2 by C by 2 by, or C plus 2 by C plus 2 by C plus 2. And we define it that way, so roughly, Wj transpose Pi minus 1 Wk is equal to our stored in Tjik. We can do this also if you use your imagination and put y and x as on the end of that. So you can also store those two. That's where the C plus 2 comes in. So we have the covariates plus those two. So you just tack them on. This means that if I want to compute that, I can compute it in one operation where I'm just using BLAS and LAPACK to perform this outer product operation, which is really nice. That's a lot to wrap your head around mathematically. So I have it graphically for you. So here we have our A transpose P0B, and that's equal to this whole thing. We have our pre-multiplied W, X, and Y. And remember, P0 is equal to this lambda G plus I inverse. So that's more or less what we've got taken care of in the middle.
Is that a diagonal matrix even with the lambda?
Yes,
Yeah.
I forgot the zeros, darn it. Okay, yes, that's a diagonal matrix of just the eigenvalues.
It's really easy to invert.
So once we've computed that, we'll store it here. And then know how we're coloring this, is those show up when we compute our P1 here using that recursive operation, which is really nice. Then we use it to compute P2 and P3. These are actually A transpose times P3 times B transpose, where A are any of A and B are any of those elements. Those matrix forms, those quadratic forms, are used in the likelihood evaluations and the derivatives. So we have to figure out a way to compute them nicely, and this works great for that. So then we're going to store that whole thing as our tensor that we're interested in. For the other matrices of this form, we have a very similar process, although the formulas are much more complicated and don't actually fit on the slides graphically. So I went with the simpler one, but it's the same process and the same storage idea. So lastly, we want to scale this to large data sets. Eigendecomposition is very key to the efficient evaluation of this. The best implementations are O of n cubed, and that's expensive. This works for small data sets, but as soon as you start getting larger data sets, you start running into problems. Biobank data has upwards of 500,000 individuals, which becomes somewhat impossible to evaluate using traditional algorithms. There are several possible ways people have tried to tackle this. One, we could use a sparse relatedness matrix. This takes advantage of the fact that most people are only distantly related and uses a threshold to induce sparsity. Second, we could only compute the relatedness matrix implicitly. There are other methods like REGENIE that do this, that reformulates the model so it doesn't have to actually compute that matrix. It trades space complexity for time complexity. It takes a good amount of time to run. Lastly, we could use more efficient hardware and more efficient algorithms. And that means we can run on high performance computing clusters to run very large eigendecompositions and algorithms that lend themselves to parallelization well. That's what we pick here. To do this, I use SLATE. SLATE is a library developed by the Department of Energy to perform optimal linear algebra. If any of you are familiar with ScalaPack, it is designed to replace ScalaPack in the near future. I use it here for efficient eigendecomposition and computing those relatedness matrices just because they're so large. It can scale to multi-node systems, not just multi-core systems, and uses MPI, the message passing interface, and GPUs if they're available. So it's really a high performance computing solution to the problem. SLATE works by storing, rather than store the entire matrix on a single process or even on a single node, we try to grid it up into these tiles. And tiles are stored across processes that are transferred to the processes that need them during parallel operations. So here's an example of a dense matrix tiled and stored across 4 processes that we refer to as ranks. Each rank only stores a subset of the tiles that are required, and those are distributed across nodes or however you decide to set up your ranks with the message passing interface. This concept can be extended to efficiently store triangular, banded, diagonal matrices, kind of whatever you're looking for. The storage methods here allow us to design algorithms that explicitly decompose the required operations into tile-level operations. Each tile operation can run on a different process, meaning once our operation graph is generated, processes are free to run the operations listed in that graph in parallel once the operation has all its dependencies completed. This allows for efficient implementations of algorithms for operations like eigendecomposition to scale massive clusters and run across tens or even hundreds of nodes. For pyGEMMA, we use SLATE to run that eigendecomposition on large data sets, and we're approaching biobank scale with what I can run. To summarize my methods, I use parallelization and shared memory to allow for fast computation over all the SNPs and keeping my memory footprint small. We saw this results in a reduction from 2 minutes from hours. So that's really nice. The tensor operations and memory layout have been used to optimize our storage and reuse values rather than throwing them away. The tensor structure in particular allows us to use BLAS and LAPACK to have fast linear algebra by aligning our memory. While I don't have an exact big O notation on this, it's slightly lower in order than what the original implementation was, just using nested for loops, and has a much lower leading coefficient, which is really nice. This becomes relevant for the types of sample sizes that GWAS typically runs with. Lastly, I run efficient eigendecomposition, which allows running on data sets that are orders larger than most methods. I'll pause briefly here to ask if there are questions before I move on to the experimental part.
Do you not have an exact O because you're working on it, or it's unclear how to get that?
It's one of those things that kind of varies. It should be like 2.87 or something like that. But it's also hard to figure that out with a parallel method because that you have to factor in a whole lot of other things like networking, speed, and stuff like that. So it's rather hard to figure out.
It's unclear what all, besides the sample size, what else needs to go in there?
Just in general, I've heard about the debugging parallel structures is kind of a difficult thing. Did you have insights into something along those lines?
Generally, the way I do it, for my undergrad, I did a lot of high performance computing stuff. So I was very lucky in GPU programming. So the way I tend to do it is you take whatever the main process is, whatever your rank 1 is, or rank 0 if you're a C user. And you get an if statement that just checks for that rank, and then that's the one that prints your errors. Everything just sends its error codes to that rank before exiting, and then that rank will be the one that outputs the error. So you don't get tons of printing, and you also get who sent that error. So it works really nicely that way.
I have a question that will reveal my ignorance, and that is this whole relatedness matrix. And if I understand correctly, you're letting the data figure out relatedness amongst everybody in the data set. It's not like you're just got families, you're comparing similarity in the.
Yes and no. Some studies do, so like I'll show a data set here in a minute, but they use mice, and with mice, you know the strain. So they like breed huge colonies and they know who's related to who.
Right.
And so they can use that to try and figure out some of this relatedness. But in human populations, we don't have all that data all the time. And so if you're running on like a 500,000 individual sample from Europe, that's going to be hard to figure out. So we use genetic relatedness because we see that large sections of the genome.
So this is like what the patients told you their genetic relatedness was, not estimating it from some genetic data?
This is estimated from the genetic data, yes. It's SNP level heritability. So it's the covariance based on that genetic information normalized over how many SNPs we use, because people who are related will have similar genetic makeup.
Yeah, I'm just, because I don't work in this area, I'm just wrapping my head about, all I can think of is all these crime shows where they say there's a 70% chance they're brothers, you know? So I'm trying to,
It's derived from those methods that are along.
so there's programs just for figuring this out from your sample of people. that
And there's a whole lot of research that went into like,
figuring
this
that out.
comes from like twin studies where they would have twins and use the probabilistic process of like, if they're this related and both twins show the trait versus only one twin shows it versus neither twin, like comparing
So there's
those.
an algorithm that's separate from your analysis that already gets this part calculated.
I wrote an algorithm within this analysis that does that, using the high performance computing stuff, because it needs to be efficient, but it does it, it's essentially just like your covariance because we code them as 0, 1, or 2. So it's your average covariance, like every SNP, there's gonna be a covariance between you and someone else until we observe that SNP.
Okay.
And so like, it's the average covariance between you and another individual across all of your genome, essentially.
Yeah,
But it's only computed once, I think
Yes.
that's just kind of the question, you get it at the beginning.
Yeah.
Which
so in my mind, I'm trying to think about all these, I don't have genetic data, but I have other stuff like, you know, microbiome, and everybody has their own version of big data sets, and I'm just trying to steal an idea if I can, but I'm not sure yet how to steal it. But I'm working on it.
Well, the good news is like, the way it works, you supply those matrices ahead of time. And so like, if you wanna just, you can fit mixed models in general by supplying different sets of matrices. And so you can give an identity matrix and a random effect design matrix.
Yeah.
And all of a sudden, you're just fitting a random effects model using
I
the software.
suspect you're getting a lot of statistical money from this trick. Because when you account for the correlation correctly,
Mhm.
you know, you have fewer false discovery rate issues, all that stuff kind of gets a little bit better. If you
Right.
take it into account.
There are a lot of studies that show power increases for
Yeah,
specifically mixed
yeah.
models of
So-
GWAS.
We'll get smarter at this, that's all I'm saying. So that was helpful, that gives me a place to think.
Anything else before experiments? Okie dokie. So I did 3 different experiments that I list here, each trying to work with something a little different. First, I used the UK Biobank to test for sample size and SNP scaling, how we perform versus other methods. I used GEMMA's mouse data set. They have a tutorial data set that was generated from a number of mice, and that allows me to check as we scale number of covariates, just because of how other methods operate. We had to keep it a small sample, but how we scale compared to them. And I used the Wellcome Trust Case Control Consortium, which is a mouthful, to show that we can replicate results from another study and interpret some of those. Our goals here are to profile the runtimes for data sets with these competing methods. So we compare to GCTA and fast GWA, just a basic linear regression I wrote in Python, and GEMMA, and demonstrate plotting functionality and then test usage in both Python and R to validate the p-values and parameter estimates. So GEMMA, like I mentioned, is an exact method for this. It gives us identical results to pyGEMMA and had opportunities for us to optimize. GCTA is an exact method that also gives us identical results to pyGEMMA, but it's extremely slow, and we'll see that in a minute. Fast GWA is an offshoot of GCTA that is an approximate method using sparse relatedness matrices and is mostly applicable to biobank data. But it's faster than pyGEMMA, but it's not as accurate. REGENIE is another exact method that I will not be showing here because it gives us the same results or similar results, but it's a 2-stage model that is much slower. For example, it took 24 hours for us to run on 50,000 samples from UK Biobank. That's a much longer time frame than what I'll show you. We're going to compare these algorithms using 2 things. One, we'll look at computational complexity of the runtimes as the parameters go to infinity. And 2, we're looking at the speed up, which is just the ratio. So you read this as like how much faster method 2 is than method 1. I'll start with UK Biobank. This was a sample of 50,000 individuals of European ancestry. We varied the samples from 50 to 10,000. SNPs from 20 to 100,000 used as our phenotype, and I didn't include any PCs or covariates because the other competing methods can't handle it for this sample size, they just take too long to run. So for each configuration, I ran it 3 times. Here are the results from the runtimes. That's a lot to take in. We're in orange and you might have a little trouble figuring out where we are because we're right here. pyGEMMA takes almost no time to run comparatively. I'll break it down by looking at the speed up plots. pyGEMMA is 30 times faster than GCTA on average. It's over 10 times faster than GEMMA for reasonable sample sizes. And about 10 times faster than just basic linear, parallelized linear regression. And while we are slower than fast GWA, I'll note, how much do you care about 30 seconds versus a minute or two when you also have to account for them using command line interface and someone who's not trained to use it, whereas this works in Python and R. Similarly, comparing as we scale the number of SNPs, about 30 times faster than GCTA on average, 10 times faster than GEMMA, about 10 times faster than linear regression. And fast GWA is 3 to 5 times faster, and that levels off as SNPs increase. But again, we're competitive given how short of a time frame we're looking at. On the mouse data set from GEMMA, this is a data set from GEMMA's tutorial, and it's 12,000 SNPs-ish and almost 2,000 mice. They're all highly related. We examined 4 different phenotypes and vary the number of covariates from 0 to 25. We use principal components for this. So here we're in orange again, and you'll see we're going pretty fast comparatively. While we still technically scale quadratically, this is what I meant by that leading coefficient is much lower, because you can see it almost looks linear, even though it's quadratic. GEMMA is certainly quadratic, and I don't know what GCTA is, but that'll just keep going up if you have to scale that further. I will also note this is 25 is kind of as many covariates as I would ever expect someone to need to run, but the fact that that makes basically no impact makes it an option that other pieces of software just don't have. Also, this is a pretty small sample size, 2,000 compared to 50,000 or something. That's a big difference.
The wide error band on the other one, because it's an approximate method, so that the runs will vary in.
GCTA is an exact method.
Oh. So
It varies because there's something in the software that makes it crash during, it randomly initializes its estimates for Newton-Raphson to start with, rather than having them in a fixed place, and I could not figure out for the life of me how to set the seeds, so that would crash sometimes. And then I'd have to, so.
The other 3 methods aren't displayed.
Fast GWA is only used for biobank data primarily. And I did not run linear regression on it, but we're still faster than linear regression. I include the speed up plot just for completeness, but it basically tells the same story. We're faster than these other methods, and these are, again, very small sample sizes. So as you increase that sample size, we get better and better. This is an example of some of the plotting function that's included in pyGEMMA. It plots very nicely using matplotlib, but for our users, it will just, you can run it through reticulate, and then it'll just save it for you. So that way there's nothing to worry about. And I also included it with all the other phenotypes that we looked at for the mice. I wanted to make sure it worked for things where there was a signal. You can see like on phenotype 2, there's a spike. And things where there really wasn't a signal, like, well, there seems to be somewhat of a signal on most of these, but something like phenotype 4, there's not really as strong of a signal, so. Finally, I'll cover WTCCC data. This was 6 binary phenotypes with 2,000 cases versus 3,000 controls in each, and almost 400,000 SNPs. I ran it with 6 PCs and compared results. Here is pyGEMMA on the y-axis and GEMMA on the x-axis. with the negative log 10 of the p-values. So exact same result here. Similarly, when you compare the beta values, you get perfect correlation as well for everything. So that's validation that it's actually working.
You happen to mention you ran it on 6 PCs. Was that a cluster or just independently?
What I mean is we included 6 PCs as covariates. This is standard practice in statistical genetics because the beta estimates tend to be so small compared to total heritability.
Like you mean principal components.
Yes.
So obviously correlation is not a measure of agreement. And I mean, it's more than that. It's right in the line y equals x. It's not just a correlation of one. It's more than that.
Yeah, it's perfectly correlated and they're the same.
Yeah, I mean, agree, you can be correlated and.
Yeah.
Yeah. That's all.
I figured I'd include both up there just so it's very clear. We're getting the same
It seems to
results.
me that the software gives you the same answers, so that's good.
Yeah,
I believe this now.
well, I presented and got that question, does it give you the same answer? And I'm like, well, it seems like it, but I haven't done the plot. So I did the plot before the next presentation. Here's an example of rheumatoid arthritis for these traits. And here are the other traits that we looked at. I will go through each in order, I guess. Bipolar disorder, we didn't see a signal, neither did the WTCCC on the right side. For coronary artery disease, there's a signal in both of them. I know their plot is small. It's hard to actually be able to compare all this, but it's in the same spot. And this is a spot that corresponds to 2 different genes. Both are important in regulating the cell cycle. So there's additional research that backs up that these are actual spots of interest for coronary artery disease. Hypertension, neither really picked up a signal. Also note the red line is after a Bonferroni correction, our significance threshold. So anything above that is what we count as significant. So we didn't really see anything for hypertension. Neither did they. For rheumatoid arthritis, there were a couple spots of interest. First is this HLA-DRB1 gene. It codes for proteins that are protein changes that have been shown to cause rheumatoid arthritis when enacted. So one of those critical pieces. Additionally, there's a spike here on 1 that both picked up, and this was an area for the gene PTPN22, which helps control immune activity in immune cells, specifically T cells.
It looks like that last one, you only had two points above your threshold, and on the original,
Right.
it seemed.
That's correct. So one thing they do, they cut theirs off at 10 to the negative 15 for their p-values. But also, studies show that you get more control over things when you lower false discovery rate when you control for the random effect properly. Does that make sense?
Does the signal, so would you say yours is representing it better than the other one?
I don't know. I mean, it's just visualization and what your choice is. Sometimes we look at what the spike is. Sometimes the top of that spike is not.
Seems like a weak spike and the other one seems like a stronger signal to me, just looking at it. That's what I would interpret it as from the visuals, and I don't know if that's correct.
Yeah, I think they're both just significance is significance. And they dig into this further, but other studies have validated this is in fact a region of interest.
Yeah, their scale, the main thing I see is their scale doesn't go all the way up to 30 plus, so they basically cut off their data.
The study's also older, and so the software available at that time had precision as a limitation, whereas mine is able to use 64-bit precision.
Oh, I see, so they couldn't get p-values that small, yeah.
Yeah, whereas I can handle up to like 10 to the negative 300. In type 1 diabetes, you see this spike at HLA-DQ-A1. This is something that has been seen to be important for other autoimmune diseases in general. So important again that we see it, they see it. And again, on that PTPN22 gene that controls for immune cell activity and T cells. We also see a spike here for type 2 diabetes, which is interesting because this hasn't traditionally been thought as heritable, but research has started suggesting that there are heritable components of that. And that shows up here on TCFL2, which can cause, depending on what mutation you have, defects in insulin secretion. All of that to say, as you scale the sample size here, the number of SNPs scales, and you scale the number of SNPs, pyGEMMA stays pretty fast. As the number of covariates grows, pyGEMMA scales much better than many of the competing methods. For large data sets, it runs properly on high-performance computing systems. For visualization and manipulation of the results, pyGEMMA is straightforward and clean. I should note I also include an interactive version with Plotly, so you can hover over your significant SNPs, find their location in the genome, and then cross-reference that with whatever else you want in an interactive way, so that's nice. And then ease of use wise, it works like any other Python or R function. If you use reticulate to pull it into R or just run it normally in Python, it works with any data that you can read from R or Python, which is important if you're not just doing human data, but like agriculture uses this. So they can use the software when they don't have the same data formats as human data, is a whole lot of other complications when you work with animals or plants. And lastly, it's available on GitHub and installs nicely with Git. I want to acknowledge the genome science training program that funded me as I was working on this. It started as a class project and spiraled into an actual project. So it's been very nice having their support. I'm happy to take any questions. I can also move it back to the first slide or whatever we need. But yeah.
There was a question online that I'll start with just because it's here. But thank you for your talk, I should say.
Yeah.
Brandy asked, did you try another multiple comparison technique because Bonferroni is so conservative?
Bonferroni is conservative. There are a number of accepted ways to do this for GWAS. You can also just do FDR correction. But there's a whole area of research into what that is. I gave that as the default. Do a Bonferroni correction. You can put in whatever you want based on whatever error correction method you would like to do. I think 1 times 10 to the negative 12, don't quote me on that, is genome-wide significant. There's an accepted threshold for what genome-wide significance is, and that could just be put in as your threshold. Yeah.
You probably get this question every time you talk about this kind of stuff, but still asking it. So the expression levels, they're not necessarily normal. And, I'm wondering if you've thought about, extending it to some kind of GEE or other dichotomized threshold version of expressions so that you don't have the results driven by a whole bunch of skewed expressions that are super high.
I guess the reason that they're comfortable making those assumptions is that even though you may not have all your model assumptions because of how normality works out, things become reasonable and we see those spikes in regions, and those have been experimentally confirmed to still work.
Yeah, I'm not too worried about missing the science exactly. And I know that you don't,
Are you talking about the Y
so I'm talking about the
or
Y.
the X?
The Y. The X is like the groups that you're trying to separate, right?
The X is your genotype, so 0, 1, or 2.
Okay.
Your Y is going to be something like height, and that might be normally distributed.
Oh. I think I misunderstood. I thought you were doing a lot with separating out these groups of diagnoses in those plots. So were those just the
In
yeah,
something like this,
yeah.
this is, so each spot in there, the chromosome is on the x-axis here. So that's a location in the genome. For each spot in the genome, we run the model 1 time.
Okay, but what is, so in the linear mixed model, what is your outcome for this particular plot? I mean, for the numbers that went into this plot.
Your outcome is type 1 diabetes status. This has to do with the infinitesimal model for statistical genetics. Because those beta coefficients are so small and that any one SNP has very little effect on the outcome, you can show that it's you don't lose very much power in doing things this way versus doing a logistic regression. There are logistic regression methods for the mixed model that exist.
Yeah.
I just haven't implemented it
So
for
it
this
really
yet.
is a zero, I was trying to set it up in my head with a continuous outcome, but you really have a 0-1 outcome.
Yeah, it works okay when you do it this way. I've compared it to doing it with the original logistic regression type approach for the binary outcome, and
Yeah,
it
no, I'm
works.
just curious, I wanna use it, I'm not trying to like break it down into like,
No, you're fine.
everybody scratching my little.
I have that
starting to
concern
fear.
too, but after running it, like seeing the same things in the same places, I
Yeah.
became convinced it has to do
Do
with.
you guys ever run like type 1 error analyses to see, if you simulate data sets that have no associations?
I did not include the null, I have a null version of it,
Yeah,
like
does it work pretty well, like?
you end up just fine, like it's along that.
It's fine, but you don't end up with like weirdness when you, okay.
It's controlled properly.
Oh,
You
interesting.
don't see inflation, and there are also inflation reduction methods that they implement here for testing on the QQ plot.
So this opens up a lot more applications in my mind because you could have a lot of things that aren't quite normal as outcomes, like
Mhm.
pseudo observations for survival data, separating out progressors, non-progressors.
But there are also other methods that do similar things that are more specifically designed. It's like, if you don't have another option, you can use this, but otherwise you can use the one that's specifically designed for that, accounting for relatedness.
I'm curious if this method was developed to only for large data sets, because I'm coming from a social science background where we don't have large data sets. So I was curious how this will perform. Like, will the model overfit because like the sample is like 500 people or something, or will it be fine?
I think with like for a GWAS style study, when you have like so many SNPs like you do here, this is like 400,000.
Yeah.
Like you start to run into issues if you have smaller sample sizes, but like, again, I was able to run on 2,000 and that was fine.
Okay.
So you just have less power because of smaller sample size. Sometimes like these effects can be really small, really hard to detect, really close to zero, even if they're positive. And so like we struggle to detect them, and that's why we need the sample sizes. It's just a mixed model. Any other questions? Yeah.
So, I mean, it seems like this is a wonderful application and why would people continue to use the other ones?
That's what I keep asking. A lot of it is people are very used to those applications. They have pipelines built on those. Like this was written by my advisor in 2012, the GEMMA original implementation. That's his dissertation. So like that type of thing exists, and they already have built around it. It's hard to convince people to adopt something new. But the pushback I've received is like, well, you're required to impute all your missing SNPs because we don't have perfect sequencing. So you have to impute it if it doesn't have a high enough score. You can't determine what their SNP is of that region for an individual. And other people are like, well, no, we can do it if you don't impute and only use the subset that are complete. And so there's debate around that, and it gets into a lot of like papers that said, well, maybe fine either way. So it's just kind of like their flavor, but also like, how do I convince a whole field to use my software?
Right, I mean, they just have to understand, it has to stand on its merits, it's got, I mean, it sounds.
And the reviewers have to accept it.
That-
Reviewers are
that's
but
it.
saying, go back and use what we know. You've got a problem.
So I have to convince people to put it in grant applications.
Is it generally mean imputations?
Yes, and like mean imputation is the way I did it for all of this, but there are imputations based on, so Human Genome Project sequence genome that's spread to like lots of other sequencing. There are now databases of large amounts of the genome where we know how correlated certain regions are with each other. So I might not have the SNP at position A, but I might have the one at 2 positions up from A. And if I have that, then those are like 90% correlated, then I can impute it. And Michigan runs an imputation server for that purpose, so people can upload their genomes and get the imputed results out.
Well, thank you very much.
Yeah.
Do you have a student that wanted to do this in November?
No, next term.
Next